So we're going to pick up where we left off. So we left off talking about the indole test. So remember, indole is an end product that we can test for. It apparently does serve some other important functions inside a cell, which is why cells actually make it. And we use the tryptone broth, which contains a lot of tryptophan. And the enzyme tryptophanase can convert tryptophan into indole. So next we're going to talk about SIM. So SIM is an agar medium that can perform 3 different tests. The S stands for H2S, or hydrogen sulfide. I is for indole, so we can use the SIM to do an indole test, but tryptone broth is more reliable because if your organism really needs a lot of oxygen to grow, stabbing it into an SIM deep is not going to be good for it, right? It needs more oxygen than that. And M is for motility, so we can use SIM to help determine motility as well. Normally in lab, we use the SIM primarily for the hydrogen sulfide test because that's one of the tests that we need to use to identify our unknowns. But you can use the SIM for indole and motility as well. But remember, it's an agar deep. So if the bacteria have either cysteine desulfurase or thiosulfate reductase, they can produce hydrogen sulfide gas. So it is a gas. So we're using an agar deep. So there's no fermentation tube inside. And you might think, well, if the agar gets cracked and broken, maybe that would indicate, right, hydrogen sulfide gas. The problem is, how do you know it's not carbon dioxide gas instead? So the agar contains iron. And so iron can react with hydrogen sulfide to make iron sulfide, FeS, or ferric sulfide technically, right? So the hydrogen sulfide gas will react with iron in the agar, right, to form iron sulfide or ferric sulfide, which is a black precipitate. So it's very easily recognizable. If the tube turns black, it's positive for hydrogen sulfide. Now remember, we can also do an indole test. And the way you did an indole test was to put Kovacs reagent into the tube. You can do the same thing here. And the Kovacs sits on top of the agar. If it turns that, hot pink fuchsia color, it's positive. If it doesn't turn that color, it's negative. And for motility, you can compare the agar and look to see if the agar is still basically clear, then there's no motility. But if the agar is cloudy, then that means the bacteria have moved away from the stab. And so that can help you to determine motility as well. So here we have 3 tubes. And so you tell me what's going on in all the tubes. So in tube number 1, where we've added Kovacs, is it positive or negative? So tube number 1, for the indole test, is it positive or negative?
Negative.
It's negative, right? What about for H2S in tube 1? Is there any black?
Negative.
Negative as well. And for motility, look at the agar in tube 1. Notice how you can kind of see through it and see the test tube rack behind it. So since the agar is clear, it's negative for motility as well, right? In tube 2, indole test, positive or negative?
Negative.
In tube 2, indole?
Positive.
Right, because we have that pretty hot pink, fuchsia layer, or red if you just want to make it easier. What about hydrogen sulfide?
Negative.
It's negative. What about motility?
Positive.
It's positive because when you compare tube 2 to tube 1, you notice how tube 2 is much more cloudy. It's not as easy to see through as it is tube 1, so that indicates motility. And tube 3 for indole.
Positive.
Positive. H2S.
Positive.
Oh yeah, right. All that black. And for motility?
Positive.
No, it's positive because the H2S is only produced where the bacteria are. So if the black is all over, that means the bacteria are all over. So it's actually easy to see motility if an organism also produces hydrogen sulfide gas because the black precipitate will be all over. But what if you had an organism that's positive for H2S but is non-motile? What would that tube look like? If it's positive for H2S but non-motile. So what would that look like?
Maybe just like a black line.
Right, so just the stab would be black, right? So just the stab, but nowhere else because the bacteria have not moved, right? So you'd have a black stab. So any questions on SIM? So we would normally do this in class with just two organisms, E. coli and Proteus vulgaris. We already know that E. coli is positive for indole, so we talked about that last time. Proteus is also positive for indole and positive for H2S, and they're both motile. So you should take a look at this. We'll just take a quick look at it and see. So it's just an article that talks about anaerobic respiration, in particular using sulfur. And so we only talked about fermentation as a type of anaerobic respiration, which just used glycolysis. But there are anaerobic forms of respiration that do have an electron transport chain. And so they do make ATP, but not as much. And because oxygen is not going to be the final electron acceptor, in this case, sulfate is. So sulfate, right, can be used. And the problem, or the reason why sulfate is not as good. There's no reason really for you to know this, but sulfate is a poor electron acceptor. It's nowhere near as good as oxygen is. And so in order for the cell to use sulfate, it actually will convert the sulfate. And in order to do that, So again, I'm not going to ask you about this, but one of the reasons why anaerobic respiration, even though there is an electron transport chain, is not as efficient, it doesn't use or doesn't produce as much ATP, is because sulfate is not very good at pulling those electrons like the oxygen does. And so the cell has to make sulfate better. And in so doing, it has to use an ATP, right, to make sulfate better. And that's sort of a general kind of explanation, which you don't really have to know. But what's the other test we use to determine if bacteria have tryptophanase? I just talked about it.
C medium.
With the tryptone broth, right, to do the indole test. How about two other methods to determine motility? When we talked about motility, what were the two other techniques we used to determine motility? Right, so when we talked about motility in lab, what two techniques did we talk about to help us determine motility?
The red, the thing that turns red.
Right, so we used motility, agar, for TTC, and wet mounts, right? Actual slides of the live bacteria to observe them to see if they were motile or not. So any questions about the SIM? So moving on to MR and VP. MR stands for methyl red and VP for Voges-Proskauer. And so the way that we do this in lab is to use just one tube of MRVP broth. And then after inoculating it and incubating it and allowing the bacteria to grow, in the next class, we would actually pour half of the broth into an empty tube. And we weren't concerned about sterility because the bacteria have already grown and made the end product. Now we need to add chemicals to see if the end products are there. And so the way we do that is by adding either methyl red or two chemicals to do the VP. Now, methyl red is a pH indicator, and so we're looking for the presence of different kinds of acids. So we say mixed acids, and these are organic acids. And the VP test is looking for a compound. Acetoin is one way. Another technical term, which is on the biochemical test chart, is acetylmethylcarbinol. So I would go with the acetylmethylcarbinol because that's what's, in the test chart. So here the test chart, acetylmethylcarbinol or acetoin is a sort of a more common term to refer to it. So in the methyl red, you simply add the methyl red. If it stays red, it's positive, right? Because methyl red is really red with acid. Once you start getting into neutral, into alkaline pH, it turns orange and then yellow. So red is positive for methyl red. The actual reagent is red to start with. It simply stays red. If it's acidic, if it's getting towards neutral, it starts getting orange, and into alkaline pH, it's yellow. With the Voges-Proskauer test, or VP for short, we have to add two chemicals called Barrett's A and Barrett's B. So students often confuse Barrett's A and B with what other chemicals that we've used?
Reagent A and B?
Right, so the nitrate reagent A and B, right, for the nitrate test. But the Barrett's reagent chemicals are different chemicals. So in lab, there would sometimes be confusion, which is why we would keep the nitrate A and B chemicals separate, and students would have to ask for them, because otherwise they would always mix up nitrate A and B with Barrett's A and B. So an important lesson in lab is always read the label on the bottle and make sure you're using the right chemicals. So the nitrates, or the Barrett's A and B, you would add 10 drops of each chemical to your tube, rotate the tube to mix, and then you had to leave it alone for 30 minutes. So the reaction was a slow one. And so 30 minutes was a minimum. And then you could examine your results. In both cases, we're looking for red color. In the case of the VP, it's kind of a cloudy red layer at the surface into the broth. Now the methyl red, if you agitate the tube, the whole tube should turn red. If you don't really agitate it, it'll just be red in the upper half. But red color is what we're looking for both of these tests. So like I said, you would inoculate the MRVP broth, incubate it. Next class, you would pour half the broth into an empty tube. Now you had 2 tubes, one for methyl red, one for VP. So glucose will be used and be converted into different organic acids. And then some bacteria can convert glucose into acetylmethylcarbinol, which we call acetoin, right, for short. And so we can test for these 2 end products, either mixed acids or the acetylmethylcarbinol. So here we have our tubes, and what's done in this picture is the 2 tubes, number 1 and number 2, are VP tests for different organisms, and tubes 3 and 4 are methyl red tests for different organisms. So let's look at tubes 1 and 2. So this is the VP test. Do you see a cloudy red layer in tube 1?
No.
No, so tube 1 is negative. What about tube 2?
Yeah.
Yes.
Right, so this kind of cloudy red layer has developed over the course of 30 minutes, and that's a positive VP test. And it is important to leave your tubes undisturbed, so don't mess with them while the color is developing, because if you agitate these tubes, the red color kind of mixes in and fades away, and then you can't see it. So we tell students just to add your Barrett's A and B, mix the chemicals together, and then leave it alone at least 30 minutes, and then carefully observe it after that time. With the methyl red test, you can add your methyl red, mix the tube, it'll stay red if there are acids present, or it'll be orange or yellow if there's no acids. Orange we kind of consider to be negative, but it could be, indeterminate. It just depends on how long you let your tubes incubate, but usually after 48 hours, if it's not red, it's negative. So tube 3 is a methyl red test. Is it positive or negative?
Positive.
Positive because it's red. And tube 4 is
Negative.
not red, so it's negative. So again, the MRVP, the only thing confusing is that students forget that they have to inoculate, then incubate, right? The tube has to grow, right? And then next class, you divide it in half. And 1 tube is for the MR and 1 tube for the VP. So any questions on MRVP? Yeah, we're not going to watch the first video because that's like 16 minutes long. You can watch that on your own. The second one, let's see if I already have that open. Nope. All right.
My name is Kevin Tokoff. In this video, we're going to discuss the theory behind the Voges-Proskauer test. All right, so let's go over some basics of bacterial metabolism that you may not have seen. We know glucose can be converted to pyruvic acid through the process of glycolysis. When we looked at the methyl red test, that pyruvic acid could be converted to mixed organic acids. And if it turned red, it was going to be a positive methyl red test. Now, the Voges-Proskauer test is looking at something a little bit different. It turns out that pyruvic acid can be converted ultimately through a series of two enzymes to a molecule called acetone. Now, acetone is not directly what we're measuring, but it turns out that acetone is involved in a series of metabolic transformations. We can form various compounds like diols or diones. Now, if we have bacteria that can produce acetone, what we can actually do is put in a series of reagents, and we can actually convert the acetone in the absence of enzymes to a dione called a diacetyl. And it turns out that diacetyl can react with peptones, specifically arginine residues of peptones, to produce a red color. Okay, so how do we do that? Well, it turns out that acetone, if we throw in alpha-naphthylamine, which we've seen before, and potassium hydroxide with oxygen gas, it turns out that acetone can be converted without the need for an enzyme into this dione called diacetyl. Now, in this mixture, we also have peptones, and peptones contain arginine residues. This is an arginine amino acid that's part of the peptone. It turns out that the arginine can react with the diacetyl, and when it does, we have a color change that is red. Now, basically, what the Voges-Proskauer test is measuring is the ability of bacteria to convert glucose into neutral end products. Okay? Diacetyl and butanediol are both neutral end products. However, we're specifically reacting these reagents with the acetone because the acetone is indicative of its ability to convert glucose to neutral end products. Okay? And a red color change would be Voges-Proskauer positive. Okay? Here's some specific results, what they might look like. Very similar to the methyl red test, if we see usually towards the top of the tube, if we have a red color, or sometimes it can go darker, a little bit purplish, that's going to be a positive Voges-Proskauer test. That means that the bacteria are able to convert glucose into these neutral end products such as acetone and diols and diones. Okay? You can see a positive result right here as well as over here. If we don't see that reddish purple color change, such as over here or on this side over here, that's a negative result. And so the bacteria do not have this ability. Okay? And also understand this because it can help you tie things together when you're tested on it. The results of the Voges-Proskauer test is red color change that we typically see towards the top is very similar to the results of the methyl red test that we looked at in another video. So if you kind of just remember for Voges-Proskauer and methyl red, you want to see a red color for positive for whatever they're measuring, then that might help you a little bit. Although what they're measuring is a little different. Methyl red was testing ability to convert pyruvic acid to mixed organic acids, so acids, whereas Voges-Proskauer is measuring the ability to convert glucose into neutral end products rather than acidic ones. Okay? And again, I'll mention this, as I've mentioned in the previous videos, this is another one of the techniques that we're going to use to differentiate between gram-negative bacillus. Okay? Make sure to watch the demonstration video on the Voges-Proskauer test.
Diagnostic microbiology and laboratory.
So you're not responsible for the biochemistry I don't really care about that.
Should we know which bacteria? Sorry.
For MVIC, yes, I'll summarize that at the end.
Okay, thanks.
So some questions. What would it mean if a microbial species was both methyl red negative and VP negative? So both tests require the bacteria to use glucose, right, to make either acid end products or neutral end products. So if the organism could not use glucose, what would that suggest about the organism? So two pathways using glucose. One produces organic acids and the other neutral end products like acetylmethylcarbinol. So if an organism cannot do either, what would that suggest about the organism? Any ideas? Is a what? Can't use glucose.
Is it an obligate anaerobe?
Mm,
Or that doesn't have to do with it?
It does have to do with that, but a non-fermenter, right, would be a possible explanation since they can't ferment glucose to make either acids or neutral end products. So the last test for MVIC is citrate. And so this is an agar slant that's used. And the idea here is that citrate is the only carbon source in the agar. So in order for bacteria to grow, they need to be able to use the citrate. And so they must have an enzyme that allows them to absorb the citrate. And that enzyme is citrate permease. So if the bacteria have citrate permease, they're able to use carbon and grow. Now the agar also contains ammonium phosphate, which serves as a nitrogen source. And so the bacteria that are able to grow because they can absorb the citrates are also able to use the ammonium phosphate. And as a result, they do produce ammonia. Now what's the pH of ammonia? What's the pH of ammonia?
11.6.
Well, so it's alkaline, right? It's just alkaline.
Yeah,
It's very basic.
strong acid.
So, right. So we are going to have a pH indicator, which is usually a bromothymol blue, which is green at a neutral pH, and at an alkaline pH, it turns a beautiful blue color. Such a pretty blue color. Really is nice. And so what we look for is green to blue as a positive test result. Here we have our two tubes. tube 1 and tube 2. So which one is positive? It's really obvious, right?
1 is positive, 2 is negative.
2 is negative, right? So again, we look for that beautiful blue color. And that tells us the bacteria have citrate permease, so they're able to absorb the citrate, use it to grow, and then they can use the ammonium phosphate and produce ammonia, which raises the pH. So this is an agar slant, so you would need to use your loop to inoculate it. And then we incubate it and look for the beautiful blue. If it's still the original green, we consider that to be negative. So for a positive test result for the citrate agar, why nitrogen metabolism is dependent on citrate metabolism?
Because of the pH levels?
No, but why is nitrogen metabolism dependent on citrate metabolism? What must the bacteria be able to do?
To produce ammonia,
No. What do they have to be able to do first?
To absorb the nitrogen?
Nope. First, absorb the citrates, right? Because it's the only carbon source. Right? So they must be able to absorb the citrate so they can grow. Then they can use the ammonium phosphate and produce ammonia, which raises the pH and the tube turns blue. But they can't use the ammonium phosphate unless they're able to grow. So they have to be able to absorb the citrates, and then they can grow and use the ammonium phosphate. So does that make sense? And then why is nitrate metabolism necessary for a positive result?
Because then you can see the change in the pH.
Yep. Right, the ammonia production raises the pH. Right, T turns blue. It means the bacteria are growing well, right? Because they can absorb the citrate. So if the bacteria have the enzyme citrate permease, they're able to absorb the citrate from the agar, so they can use it as a carbon source to grow. And because they're now able to grow, they can use the ammonium phosphate and produce ammonia, which raises the pH and turns the agar blue. Any questions on that? So for urea hydrolysis, or the urea agar, or the urease test, urea is the substrate. The enzyme that converts urea to ammonia is urease. And we just saw that ammonia has an alkaline pH, so it's a pH change. So we can easily observe that. In this case, we are going to be using phenol red as our pH indicator. What color does phenol red turn with different pHs?
Pink, orange, yellow.
Right, so what does yellow mean?
It's basic.
Yellow?
Oh no, yellow means it's acidic.
Right, as we saw with carbohydrate fermentation and the MSA plate, right? And what color does phenol red turn with base, with alkaline pH?
Pink.
Right, that hot pink fuchsia color. So you can look at a urea agar tube and tell me what the pH is, right, by the color. So here we have 3 tubes, right, tube 1, 2, and 3. Although the 1, 2, and 3 numbers here got offset, they should be over here. But tube number 1 is yellow. Is that positive or negative for urease, and what's the pH? Is it positive or negative?
Positive.
It's yellow?
The first
Ammonia?
one?
Yeah,
Negative.
it's negative because we're looking for ammonia production. And so what's the pH?
Acidic.
In tube 1, it's acidic, right? Oops. In tube 2, is it positive or negative? Of the middle tube, tube 2, is it positive or negative? And this tube here, is it positive or negative?
Positive.
It's positive. It's fuchsia. And so what's the pH?
Alkaline.
Right. Or basic, right? And tube 3, is it positive or negative?
Neutral.
No, but is it positive or negative for urease?
Oh.
It's not fuchsia,
Negative.
so it must be
Negative.
negative, right? And of course the pH, as you said, neutral. Right, so if the agar is still the original peachy orange color, it's negative, and the pH is around neutral. If it turns hot pink, right, the fuchsia color, it's positive and has an alkaline pH. If the tube is yellow, it's negative, right, for urease, and has an acidic pH. Any questions on urea, agar? So where do we find urea?
In urine.
In urine, also in sweat. So there's urea in sweat as well, but urine is what we typically think of. And so what happens if you let urine sit, right, for a long time? So what happens if urine sits for a long time, like 24, 48 hours? It starts to produce what?
Will it produce ammonia?
Ammonia, right? Because there's always bacteria around, and many bacteria love urine, right? They use it as a food source. And so if urine sits around long enough, it produces ammonia. Ancient peoples discovered this, and so they would use the ammonia as a bleaching agent. And so all the people in a village would all pee into a large container, let it sit for several days, and then they could use that to soak their animal skins and make them lighter and whiter. And because the ammonia was a bleaching agent, some women said, hey, look, we can use this. And they would use things like camel urine and put it on their hair to make it lighter. And so that was the beginning of bleach blondes using urine, right? Because when you bleach your hair blonde, you're using an ammonia product, right, to do it. And so the first ammonia came from urine, and that was used for bleaching things to make them lighter and whiter. Any questions? So we would have tested in lab E. coli and Proteus, to see which one was positive for urea. So what causes the yellow color? Easy question, right?
Is it the ammonia?
No, yellow color. Yellow is acid, right? Yellow acid, pink is base. And then we've talked about Helicobacter pylori in lecture when we had the lecture on bacterial groups. And let's see, do we have? And so remember we said that Helicobacter pylori is an organism that many people have in their stomachs, and most of those people do not suffer any problems from it, but some people do. And what were some of the problems they went on to experience?
Ulcers.
Ulcers, right? And then severe cases of stomach cancer. So a common cause of ulcers is Helicobacter pylori infection. So of course you get that burning pain from the ulcer. You may have nausea, loss of appetite, infrequent burping, bloating, unintentional weight loss, because if eating causes pain, you tend to eat less. But it can progress and be more severe, ultimately potentially leading to stomach cancer. But it just depends, right? Most people will never have any symptoms of a Helicobacter pylori infection. Others may get ulcers, and some others, a small percentage, it can lead to stomach cancer. So how do you treat? Well, if you're producing lots of acid, you can use proton pump inhibitors like Prilosec or Nexium. And so they actually reduce the amount of acid produced in your stomach. And that can help to alleviate symptoms and give your ulcer time to heal. But a problem with the proton pump inhibitors is that we need stomach acid to absorb certain nutrients like calcium. So it's very important. And if you take a lot of proton pump inhibitors, you're not absorbing as much calcium as you should be. And so that can lead to osteopenia and then osteoporosis. So you're normally only supposed to take a proton pump inhibitor for like 6, maybe 8 weeks and then stop. But it depends on your condition. Some people basically take it every day, forever. Others just take it for, 6 to 8 weeks and then they can stop. Every once in a while, they may have to take a pill here and there, to deal with heartburn. But you're really not supposed to take proton pump inhibitors every day for the rest of your life because it does inhibit the absorption of important nutrients like calcium. Now, interestingly, histamines can actually help to reduce stomach acids as well. So we take histamine blockers. So we take antihistamines for allergies, but certain antihistamines can also reduce stomach acid. So like a cimetidine, which is a brand name Tagamet. So that can help to reduce stomach acid. Or if you're familiar with Pepto-Bismol, bismuth subsalicylate can help to protect your stomach, to reduce the acid that's already been produced. So there are certain treatments, and of course, antibiotics. If you truly have a Helicobacter infection, then antibiotics can be used to treat that. But you just need to be careful with your proton pump inhibitors. They're not meant to be taken forever. If you already have osteopenia, where your bones are thinning, your proton pump inhibitors can cause osteoporosis. So you should be supplementing with calcium and vitamin D supplements. Now, what's weird about Helicobacter, as I've said, many people have Helicobacter. It causes no problems. In some people, it could lead to stomach cancer. Exactly how or why, they don't know, but it's a relatively common organism. So there is some evidence showing that Helicobacter infection can cause, stomach cancer. There's also some evidence suggesting that Helicobacter may reduce the risk of some cancers. So we just don't know. But it could be that a long time ago, humans were colonized with Helicobacter, and it actually was beneficial. But as our world changed, as our environment changed, our lifestyles, our diets changed, having Helicobacter was no longer beneficial. Or the use of antibiotics may have really screwed up, Helicobacter colonization. So we just don't know.
Yeah,
yeah.
Sorry. Go ahead. Sorry.
Well, no, you can go ahead.
I was just gonna say, I interned with genetic counselors, like 2 summers ago, and they were on a team dealing with hereditary diffuse gastric cancer, which is there's a genetic mutation that causes that type of cancer, and they found in one study that patients who had the genetic predisposition for the cancer, but who were infected with H. pylori, never got the cancer.
Right, so there may be a protective benefit if you are genetically predisposed to develop stomach cancer. So again, we just don't know, right? There are many variables involved. Humans are terrible test animals because we're not the same genetically. We don't eat the same foods. We don't live the same lifestyle, right? We're exposed to different environmental toxins. And so there are many, many variables, to take into account. So again, H. pylori may be protective, may be harmful, may do nothing at all. It just depends on you, your genes, your environment, your diet. And so We just don't know. But it is interesting that in some people, H. pylori could lead to stomach cancer. In others, it could protect them. So, of course, we just saw ulcers, stomach cancer. Don't worry about how it's related to Proteus. We don't care. Yeah, don't worry about how H. pylori protects itself either. So, and here's a couple answers to the MRVP questions. Again, having to do with the idea that some bacteria, if they're non-fermenters, are going to be MR negative and VP negative. So that's included here. Like I said, don't worry about these other two questions about Helicobacter. We don't care about that. So I do want to add one more thing, and it does have to do with MVIC. And so in class, we do want you to know the results of E. coli and Enterobacter aerogenes for MVIC. So, and it's very easy to remember. E. coli is positive for indole, positive methyl red, negative EP, negative citrate, plus, plus, minus, minus. Enterobacter is the reverse. Enterobacter is negative for indole, negative methyl red, positive EP, and positive citrate, plus, minus, minus, plus, plus. So you should remember the test results for E. coli and Enterobacter for MVIC. So any questions on the biochemical tests? Okay, now we're going to talk about streak plate. So there's a handout for the streak plate. So we have talked about streak plate in lecture. It's a very important technique. So important, normally we would grade you on making a streak plate. So you would actually have to make one, and it would have to be good. Obviously, we're not doing that right now. So you'll just watch a video to see the technique and learn what is a good plate and a bad plate. But whenever you get a sample from a patient or the environment, it's going to be mixed. So you need a way to separate those mixed bacteria. And the streak plate technique is a good way to do that. And when you do the technique, it's basically a dilution on the surface of the agar plate. And we'll see how that works. But if you do the technique right, you will separate those bacteria and they will form discrete, isolated colonies. Then you can take one of those colonies and use it to make a pure culture. So the streak plate technique is very important. There's more than one way to do it. We're going to just look at the most common way to make a streak plate. You can do it using either 3 streaks or 4 streaks. Generally speaking, if you're using a broth culture, 3 streaks is better. If you're using an agar culture, then 4 streaks. And the difference is, if you're using an agar culture, the inoculum, the amount of bacteria on your loop is a lot more to start with. And so 4 streaks may be necessary to get good separation. But in a broth culture, you're starting off with a lot fewer bacteria. So 3 streaks is fine. 2 important things to remember. You're only using 1 loopful of bacteria, and that's it. Just 1 loopful. And you need to flame the loop in between each streak. So you only put 1 loopful of bacteria on the upper portion of the plate, and you're going to flame your loop each time you streak. And the idea is to grab cells from the previous streak and then spread them out. Oh, geez. This thing jumps around when it's got pictures in it. So here we see a good streak plate. This first zone here is zone 1. So this is your one loop full. You put them into zone 1, and that's it. No more bacteria. You flame your loop. And then you're going to drag your loop into zone 1. Here they're just showing it one time because they used an agar culture to start with. If you're using a broth culture, you want to go into zone 1 like 3 or 4 times and then stay out of it and spread them out here. Then you flame your loop. Again, you drag it into zone 2, usually 3 or 4 times, and then you stay out of it and you spread out the cells here. So this is zone 3, zone 2, and zone 1. So you notice we have a lot of bacteria in zone 1, fewer bacteria in zone 2, and the fewest in zone 3, where we have nice, discrete, isolated colonies. So let's watch a video.
In this video, you will learn how to perform streak plate technique. The streak plate method allows isolation of a single colony from a bacterial culture by splitting the plate into quadrants and diluting the bacteria repeatedly as the loop is streaked through each quadrant. To light a Bunsen burner, start by closing the valve at the base by rotating the collar so that the vents are covered. Then slowly turn on the gas. Once you hear the gas, ignite the burner and set the height of the flame by adjusting the amount of gas. The temperature of the flame can be adjusted by rotating the collar to control the flow of oxygen. The coolest flame is yellow and orange. A medium flame is blue, and the hottest flame is a roaring blue flame characterized by a clear blue cone in the middle. The tip of the cone is the hottest part of the flame. Flame an inoculating loop to sterilize and allow it to cool. You can lightly touch the loop on an empty spot on the bacterial plate to make certain the loop is cool. Gently scrape a colony from the surface of the plate so that it collects on the loop. Transfer the bacteria to your LB agar plate and gently rub the loop back and forth across the top left corner of the plate about 10 times. Stay in the top left quadrant of the plate and do not break the surface of the agar. When finished, flame the loop. Rotate the plate 45 degrees or a quarter turn and draw the loop through one end of the first streak and rub back and forth in the second quadrant about 10 times.
Now you notice that what he did, he used the loop, he moved into zone 1 several times, and then he stayed out of zone 1 and spread the bacteria into zone 2. And what you're going to notice after he flames his loop, he's going to rotate the plate again 1/4 turn. He's going to drag the loop into zone 2 a few times and then stay out of it and spread out those bacteria.
Flame the loop again to sterilize. Rotate the plate 45 degrees and draw the loop through one end of the second streak and rub the loop back and forth in the third quadrant about 10 times. When finished, flame the loop again. Repeat the process one final time.
So you're going to see
Rotate
Observe
the plate
carefully
45 degrees and draw the loop through one end of the second streak.
how he
and
goes
rub the
into the
loop back and
previous
forth
streak
in the third
sometime,
quadrant about
several
10
times, and then he stays out of it, right, to spread out those bacteria.
When finished, flame the loop again. Repeat the process one final time in the fourth quadrant.
So into the previous streak and then stay out of it.
Flame the loop after completing the streak plating. Invert the plate and place it in an incubator at 37 degrees Celsius for 24 to 48 hours. After 24 to 48 hours, the colonies will have grown and can be examined.
And they're not going to show us their plate. But we have a video here which is going to show a plate that's incubating. So we're watching the bacteria on a streak plate. So as they grow, you see zone 1, which had the densest growth, that appears first. Zone 2, and they really didn't get any growth in zone 3. This probably represents the growth in zone 3. But what we're seeing are the colonies develop. And so zone 1 has the most, zone 2 has fewer, and then probably here they did zone 3, but they only got a few here, right? So they're just grabbing, using one streak to pull bacteria. And so they got some here. Now, this organism is Bacillus cereus. And so what do you know about Bacillus?
It has spores.
Right, it makes spores. So we talked about where was the best place on an auger slant to find spores. So where was the best place on an auger slant to find spores?
Was it on the top?
Right, the top half. So in an auger plate like this, where would be the best place to find spores? Where would you go to get your sample to make your spore stain?
Probably from the first streak.
Yep, in zone 1, in fact, in the middle of zone 1, right? Because that's where the thickest, right, densest growth is. So that's where conditions are going to deteriorate first, in the middle of zone 1. So that's where you get your sample, right, to look for spores. If you took it from the edge of zone 1, well, that's where the healthier bacteria are, right? Because they're growing and spreading outward. And a colony, again, these are young, healthy bacteria. So you would not find spores here unless you let the plate sit for a week. But after 2 or 3 days in the middle of zone 1, you'll start to see spores developing. So that's where you would have looked, for spores on your second unknown, is to get them from the middle of zone 1. Now, in this video, what we're looking at is the formation of a colony. And so it's just showing you the bacteria which are multiplying. All right, so they start out and they slowly multiply, spread out, and eventually there'll be enough cells to form a visible mass that we can see, and that's our colony. And so notice how in the middle here, it's thickest and most dense with bacteria. So over time, this is where spores will start to develop, right? But if the colony is small, it's still too young. You'd have to wait for the colony to get old. And then you'd look in the center here, and that's where you would find spores. You can see how much thicker and denser it is in here. So what I would normally do in class is I would first talk about how to make a streak plate and draw it on the board, right, illustrating the technique. And then I would actually demonstrate the technique and make a streak plate while students were watching. And then you guys would go back to your desks and you would practice with pencil and paper, right, to get the general idea before you actually made a streak plate. Because you would only get one plate to use. And so if you mess that one up, you'd have to wait till the next class, and then we see how bad it was. And then I would give you a second chance, right? A do-over. And if the second plate was better, that's the grade you got. Some instructors, what they do is they said, okay, I'll give you a second plate, but whatever result you get on the second plate, that's the grade you get. If it's worse, then you get the lower grade. Right, so I was much nicer on that. So here we have two examples of bad plates, bad streaks. And so in the first plate here, what probably happened is the student did not flame the loop in between each streak. And so there's still lots of bacteria on the loop. And so they get lots of growth in every zone and no separation. In the second plate here, they weren't following the proper technique at all, right? And this is actually, it happens every semester in every class. You know, even though I show it on the board, I physically demonstrate it, the students practice with pencil and paper, when they actually make the real plate, they don't follow the proper procedure. So somewhere there's a disconnect between their brain and their hand. And so, right, nothing was done correctly here. So here are some good plates. Right, the first two plates are using 4 streaks. Notice that they go into the previous streak multiple times and end up with some good separation here. This plate here is actually one of our plates, and we just use the 3-streak technique, but we got good separation here in zone 3. Right, so you want to have multiple discrete isolated colonies in the last zone. So that's the goal. So next we'll have some pictures of some plates, and you can tell me if it's good or bad and what makes it good or bad. So plate number 1, is that good or bad?
Bad.
They don't
It's
overlap.
bad, right?
Bad, yeah.
So why is it bad?
The zones looks the same. Not different.
So it looks like maybe they used 3 loopfuls of bacteria, 1 for each zone, because the zones don't really appear to be connected to each other, right? So they just took 3 loopfuls, 1 loopful, another loopful, and another one maybe. What about plate number 2, good or bad?
Bad.
Yeah, bad, it's even worse, right? Clearly, there's no connection. They're 3 separate zones, right? And again, students do this every semester, right? Even though the instructions say, even though I say, even though I demonstrate 1 loopful, they grab 3 loopfuls, 1 for each zone. What about plate number 3?
Bad.
Bad?
The first 2, well, that means that's 4 zones. 1, 2, 3, 4.
I think it's good because there are single colonies.
Yeah, it's a good plate, right? We ended up with our isolated colonies in the last zone, so this plate is perfectly good. What about plate number 4? Good or bad?
Good.
Good? Why is it good? Remember, you should have multiple colonies in the last zone. They don't have multiple colonies. And it also looks like they just did 1 pull, from zone 1 and 1 pull from zone 2, and that was not enough, right? So we normally recommend you go into the previous zone at least 3 or 4 times, right, to grab enough bacteria to separate. And what about plate 5?
Bad.
It's bad, right? So they put the bacteria in zone 1, but then they missed, right? They didn't actually go into zone 1, they missed, and so they didn't grab anything for zone 2. And since there was nothing in zone 2, they couldn't grab anything and spread in zone 3. So, but these are all common examples of problems that happen when you do a streak plate. And there's really no way to know for sure if your plate is going to be good until it's incubated and the bacteria grow. Because even though I've been doing this for decades, when I demonstrate, most of the time my plates are good, but sometimes they're not good. So it's not something that you can do perfectly every single time, but we want you to learn how to do it properly. And so we would let you do it once and then give you a second chance. Because for unknown number 2, one of the first things you had to do was make streak plates, right, to create pure cultures of your organism so that you could then test that. And so we wanted you to get some practice first making streak plates before you had to make them to use for your unknowns. But it is something that really does require, practice and doing it more than once or twice. But we've seen some good plates and some bad plates. So, any questions on the streak plate technique? Any questions on the biochemical tests? Right, so make sure you study this chart. Yeah, question?
Yeah, for the unknown, we'll have to identify something based on the biochemical tests.
Yes, I'll talk about that on Monday because I am going to be emailing you your test results. So there'll be pictures. You just have to interpret them. And there are flowcharts to follow, right, to identify, and then a report sheet. So we'll go over that on Monday. It's nothing to worry about because it's much simpler than what you actually would have done in class, because in class, you would have done all the work, all the inoculating, right, interpreting tests. And some of you would have contaminated your results, so that would cause some problems. But I'm just going to be giving you pictures of test results for you to interpret and follow the flowcharts. So much simpler than what you would have done in class. So no reason to worry about it. And I haven't decided for sure if I'll make it due Wednesday or Friday, but it will be due next week because you're not actually doing any work except to look at pictures. So any other questions? All right, that's it then for lab. You too, Sarah. So enjoy the weekend. It's supposed to be nice. Keep your fingers crossed, for Joe Biden. We'll see what happens.